Good evening to all the students. So yes, we are in our part 3 of our biomolecules, which is a major chapter from cell biology. So far we have completed what concepts, ma? From the beginning, Raghav? So far we have dealt initially with biomacromolecules, firstly about elemental analysis, chemical analysis, ash analysis, okay. After that we have looked into the biomacromolecules. Firstly we have completed carbohydrates and then proteins. Once you briefly tell me about the carbohydrates classification. We have discussed about monosaccharides, disaccharides, oligosaccharides and polysaccharides, right ma? And then in proteins we have discussed about 4 structures, 4 levels of protein structures. What are they? Primary structure of protein, secondary, tertiary and quaternary structures. Okay, and then we have started with nucleic acids. Of nucleic acids we have begun with nucleotides. What will be the nucleotide composition, ma? Nitrogen base, sugar and phosphate group, right? Nucleoside composition will be sugar and nitrogen base. Very good. So what type of sugar is present in RNA? Ribose sugar. Is OH group present at second carbon of the sugar? For ribose it is present. Very good. So this and all we have learnt, right? To which carbon of the sugar is nitrogen base attached to? To which carbon of the sugar? To which carbon of the sugar nitrogen base is attached to? Aisha, can you try? To which carbon of the sugar nitrogen base is attached to? No. Simple question. Let us assume this as a ribose sugar, okay? C1, C2, C3, C4 and C5, okay? To this at first carbon nitrogen base is attached. Fine. If the nitrogen base is purine, purine is how many? Purine has how many nitrogens? 9, right? Purine has how many nitrogens, ma? 9. So C1 of the carbon is connected to N9 of purine. Examples of purine? Adenine and guanine. Maintain the running notes neatly. Pyrimidines includes what, ma? As it is ribose sugar, we have what? Cytosine and uracil. Thymine will not be present, right? So C1 to the C1 of the ribose, N1 nitrogen will be connected from nitrogen base. What are the nitrogen bases of pyrimidines? Under pyrimidines we have cytosine and uracil for RNA, okay? And to which carbon phosphoric acid is attached, that is phosphate group is binded? C5. This makes out one nucleotide. At least now you register in your mind. Huh? Yeah. Now we will look into the linkage of nucleic acids. We are clear with the nitrogen bases, okay? Now we have to look into how a nucleotide chain is formed. Fine. What is a nucleic acid? Polynucleotide chain. The building blocks of the nucleic acid is nucleotides. How the nucleotides are connected to each other. So third carbon, at third carbon both in RNA and DNA, OH group is present, yes or no beta? OH group is present in both ribose and deoxyribose. To this phosphate group which is present at 5th carbon will be connected. So here you see once, first you listen, at 5th carbon phosphate group is attached, right? This is through what bond, Nana? Phosphoester bond. Phosphoester bond. Fine. C3 carbon of one nucleotide is connected to, C5 carbon of one nucleotide is connected to C3 carbon of the preceding nucleotide. Okay, so here 2 bonds are involved, 2 ester bonds are involved, right. So this is called as what? Phosphodiester bond. Okay, phosphodiester bond. You should be very clear on the linkages of forming a single nucleotide, linkages of forming a polynucleotide chain, and you should be clear with the bond that is connecting 2 nitrogen bases in forming a double helical structure or double stranded nucleic acid or DNA. So here you look into, take down clearly. First, for the formation of nucleoside, what bond is involved, ma? N-glycosidic bond. N-glycosidic bond. Fine. Second is for the formation of a single nucleotide. For the formation of a nucleotide. Okay. So here for the nucleoside, what all are present in a nucleoside? Sugar and nitrogen base. Yes or no, ma? To this, what is connected? A phosphate group is formed. So in a single nucleotide, what are the bonds that are involved in a single nucleotide? Tell me, what are the bonds included in a single nucleotide? One is N-glycosidic bond. N-glycosidic bond. Second bond will be phosphoester bond. Phosphoester bond. Fine, right? Now, third is the bonds which are involved in the formation of a polynucleotide chain. Polynucleotide chain. 3 bonds are included. One is N-glycosidic bond, second is phosphoester bond, and third bond which is involved over here is phosphodiester bond. Phosphodiester bond. Clear, right? Now, lastly, what are the bonds that are involved in the formation of a double stranded DNA? Okay. Fourth is bonds involved in the formation of double stranded DNA, that is in the double helical structure. 4 bonds along with these 3, N-glycosidic, phosphoester, phosphodiester bond. Along with this, hydrogen bond is involved. Hydrogen bond is formed in between 2 nitrogen bases of 2 nucleotide chains. Okay. Sorry, in between the nitrogen bases. Yes, in between the nitrogen bases of 2 nucleotide chains. Okay. So double stranded DNA is one more bond is involved, that is hydrogen bond between 2 nitrogen base. Yes, nitrogen bases of 2 polynucleotide chains. As you said, it will be in between purine and pyrimidine. That is, adenine pairs with thymine, cytosine pairs with guanine. So, are you clear with linkages in the nucleic acid, beta? Okay? Write neatly, Raghav. Yeah. So, you tell me, for a nucleoside, what is the bond involved in the formation of nucleoside? Good. For nucleotide, 2, N-glycosidic bond and phosphoester bond. For a polynucleotide chain, phosphodiester bond is also included. Double stranded DNA, along with these 3, hydrogen bond is also included in between the nitrogen bases, of 2 polynucleotide chain. Okay. 2 hydrogen bonds are present in between adenine and thymine, 3 hydrogen bonds are present in between guanine and cytosine. Now we will look into the Watson and Crick's model of DNA. Okay. Double helical model proposed by Watson and Crick. Double helical DNA model proposed by Watson and Crick. The most popular question from this model is, what are their propositions based on? Okay, their propositions are purely based on base pairing. Okay, Watson and Crick's model of double helical DNA, their propositions are purely based on what, ma? Base pairing. Base pairing. Base pairing means what? Nitrogen base pairing. Okay, in between purines and pyrimidines. Okay, adenine and guanine, these two are purines, right? Adenine pairs with thymine by 2 hydrogen bonds. Guanine pairs with cytosine. Guanine pairs with cytosine by 3 hydrogen bonds. Fine. This is the first thing. Next, the most stable form of DNA is B type DNA. Okay, wherein each pitch, that is one turn of this double helix, length will be 3.4 nanometers. 3.4 nanometers is 34 angstroms. This is one pitch or one turn. One pitch or one turn is from here to here. One pitch or one turn will have 10 base pairs, 10 base pairs, and length will be what? Yeah, 34 angstroms or 3.4 nanometers. Clear, right? And distance between two base pairs, distance between two base pairs is 0.34 nanometers is the distance between two base pairs. So, clear, right? What is the distance? 0.34 nanometers is 3.4 angstroms. You have to be very clear with the length of DNA. Okay, in one pitch or one turn, what is the length? 34 angstroms or 3.4 nanometers. It is having how many base pairs, beta? 10 base pairs. Distance between two base pairs is 0.34 nanometers or 3.4 angstroms. Any doubt anywhere till here? No, right? And here in this double helical model, towards inner side, listen carefully, towards inner side, because of this hydrogen bonding, nitrogen bases are phased towards inner side, and towards the outer side, sugar phosphate backbone is formed. Okay, so DNA double helix is formed by what? Sugar phosphate backbone. Sugar phosphate backbone, where nitrogen bases are phased towards the inner side. Fine, nitrogen bases phased towards the inner side. Next is two strands of this double stranded DNA, these two strands, they run anti-parallel to each other. Okay, two strands are anti-parallel to each other, and nitrogen bases which are connected at the center, these are complementary to each other. Okay, so two things you have to make a note here. One is two strands of this DNA, two strands of the double helical DNA are anti-parallel to each other. Anti-parallel to each other. One runs from 5 prime to 3 prime and the opposite strand from 3 prime to 5 prime. Okay, so this way. And complementarity between nitrogen bases. Complementarity between nitrogen bases. So, you are clear with these points, no? Most important points, beta. Let us recall once quickly. First thing is, the hallmarks of their propositions is what? Base pairing. Base pairing is the hallmark for Watson and Crick's model. Okay, wherein, and the second point is sugar phosphate backbone. Third point is two strands run anti-parallel to each other, and fourth is the complementarity between the nitrogen bases, A to T and G to C. And then one pitch or one turn of DNA will have approximately 10 base pairs, with the length of 34 angstroms or 3.4 nanometers. Distance between two base pairs will be 0.34 nanometers or 3.4 angstroms. So, till here it is clear, right? Yeah. Next we will move on to different forms of DNA. Okay, there are many forms of DNA, but as of now we have to be clear with three forms of DNA. One is aDNA, bDNA and zDNA, of which most important model is B form of DNA, which is the most stable form of DNA. Okay, look into the differences clearly beta. First is helix sense, how they are folded. aDNA is right-handed helix. Listen carefully, aDNA is right-handed helix. I will send you the PPT anyhow. Okay, listen carefully. aDNA is right-handed helix, bDNA is also right-handed, zDNA will be left-handed helix. Okay, base pairs per turn, per one turn or one pitch. Per turn or pitch of the DNA is one and the same. Okay, here you will be having 11 base pairs per one turn. Here 10.5, so we can consider it as 10 base pairs per turn, and Z type of DNA is having 12 base pairs. Major groove and minor groove. Major groove is narrow and deep for A form of DNA. It is wide and deep for B form of DNA. Flat or absent in Z form. Can you see? It is flat or absent. Major groove is not present. Minor groove is wide and shallow. Here it is narrow and deep in B form of DNA. And narrow and deep in Z form of DNA. Minor grooves you can see, but no major grooves are present. And then occurrence, if you see, dehydrated DNA. This A form of DNA you can see as dehydrated DNA, where very low water content is there. But here in normal physiological DNA, in human DNA will be in what form? So it is B form of DNA, normal physiological DNA. Whereas in high salt concentration or special sequences, GC, such sequences when they are there, it will be Z pattern of DNA. Next, most common in all cells is B form, these are in the less form. Stability also, most stable form of DNA is bDNA, these two are least stable. Okay. Once go through the differences clearly for 1 minute, and then we can move on to the Chargaff's rule. Okay. Yes. Here if you see Erwin Chargaff in the year 1950, he has proposed few propositions. First is, in a double stranded DNA, the amount of purines will be equal to the amount of pyrimidines. That is, amount of purines A plus G will be equal to amount of pyrimidines T plus C, thymine and cytosine in a double stranded DNA. The amount of adenine will always be equal to amount of thymine. Amount of guanine is always equal to the amount of cytosine. As such, if you can take 100% of nitrogen bases are present in a double stranded DNA, when it is given like 25% of cytosine is there, then you can easily identify how much percent of other nitrogen bases are there, yes or no. If 20% of cytosine, so 25% of guanine will be present. Rest how much? 50% will be there. Of 50%, adenine and thymine both will be equal, right? 25% of adenine will be there and 25% of thymine will be there. As such, you have to calculate. I gave you a very simple example, but applications will be in this mode only. Okay? And then the base ratio is nothing but that is the ratio of adenine, thymine by guanine and cytosine. This will vary for each species beta. For example, if you take Escherichia coli, E. coli, those are prokaryotic cells. Their base ratio will be less than 1. For example, in E. coli, base ratio will be less than 1, that is around 0.9. Whereas in human cells, eukaryotic cells, base ratio will be base ratio will be 1.33. Clear, right? Any doubt anywhere? Next, the deoxyribose sugar and phosphate components occur in equal proportions. Sugar and phosphate are also in equal proportions. So, once again recall quickly what are the Erwin-Stargoff's propositions. First is amount of purine will be equal to the amount of pyrimidine, amount of adenine always equals to thymine, guanine always equals to cytosine. Base ratio, that is A plus T by G plus C, will vary amongst the species. In E. coli it is less than 1, in human cells the base ratio will be more than 1. The deoxyribose sugar and phosphate components will always be in equal proportions in a double stranded DNA. You have to be clear with Watson and Crick's model beta. After that, Erwin-Chargaff's propositions. Okay. Next, types of RNA. Before knowing the types of RNA, you should be clear with the synthesis of RNA. Where is RNA actually synthesized? So, here if you see cytoplasm and nucleus at the center. Okay. In the nucleus and nucleolus at the center. So, consider this as nucleus. Inside a nucleus, this will be the nucleoplasm, nucleus. This entire thing will be nucleoplasm, and the small area, let us assume this as nucleolus. Fine. Inside a nucleolus, you can find RNA polymerase 1. RNA polymerase 1. This RNA polymerase 1 is helpful in the synthesis of rRNA. Fine. In the nucleoplasm, we have RNA polymerase 2 and RNA polymerase 3. RNA polymerase 2 and RNA polymerase 3. RNA polymerase 2 will synthesize mRNA or messenger RNA. RNA polymerase 3 will synthesize tRNA or soluble RNA or adaptive RNA as well, because it helps in the transfer, it helps in the making of amino acids, okay, from triplet codon. So, here you see the functions and the proportion in the total cellular RNA. In total cellular RNA, what is their proportions and what are the functions of this? Messenger RNA, this carries genetic code from the DNA to the ribosome. Okay. So, mRNA concentration or the proportion amongst the total types of RNAs will be 5%, and then ribosomal RNA which plays both structural and enzymatic role in the ribosome, which is of 80%. So, the more proportion is occupied by what beta? Ribosomal RNA. Last is tRNA. This will help in the transfer. This transfers the amino acid to the ribosome during the translation process. This is around 10 to 15%. Amongst total, this is around what? 10 to 15%. Till here it is clear? Any doubt anywhere, ma? No, right? So, as such you can find mostly it is single stranded RNA only. Double stranded RNA cannot be formed because it is not stable. Fine? Because of the free oxygen group which is present at the second carbon of the ribose sugar. Fine? Any doubt anywhere? Clear? Yeah. So, with this we have finished about the nucleotides beta. Now, the last biomacromolecule, that is lipids. Before going to lipids, we have to have one clear idea. Once again, let us recall that in acid soluble fraction and acid insoluble fraction, where we got two fraction after chemical analysis, lipids are obtained in acid soluble pool or acid insoluble pool? Acid insoluble pool or retentate. Very good. Lipids are obtained in acid insoluble pool or retentate. Fine, they are obtained in acid insoluble pool or retentate. So, here you will find all the biomacromolecules, yes or no, of that lipids are also found. But all the other biomacromolecules which are obtained in acid insoluble pool or retentate, their molecular weight is around 800 daltons to 1000 daltons, yes or no, ma? their molecular weight is around 800 daltons to 1000 daltons. But then lipids molecular weight will be, how much? Lipid molecular weight will be less than 800 daltons. It is less than 800 daltons. Yes. Why is it less than 800 daltons? Because at the time when leaving tissue, any tissue is grinded with mortar and pestle, the cell membrane get ruptured. Cell membrane is majorly composed of lipids, yes or no? So, those lipids will form what? Micelles, like this. Micelles will be formed. Okay? Cell membrane gets ruptured and micelles will be formed. As such, they are accumulated in the acid insoluble pool, but then their molecular weight is less than 800 daltons. Yes. Once just go through these points, okay. Just take down those points and now I will be starting it. So, here there is a comment. Okay, fine ma, just go through the class. Any doubt you are having, you can drop a message in the comment box. So, yes, lipids are obtained in the water insoluble pool, but then their molecular weight is less than 800 daltons, right. Now, see, we have 3 different types of lipids, 4 different types of lipids. One is simple lipids, second is conjugated lipids, and third will be derived lipids. Firstly, lipid is a combination of fatty acid and glycerol. A fatty acid will have carboxyl group. This is attached to the R group. Okay, fatty acid will have carboxyl group, right? This is attached to a functional group. This forms out to be a lipid. So that R group can be anything. It can be a methyl group or an ethyl group or higher number of CH2 groups. For example, palmitic acid, R group is having 16 carbons, including carboxyl carbon. Arachidonic acid has 20 carbon atoms, including carboxyl carbon. Including carboxyl carbon, palmitic acid is having 16 carbons. Arachidonic acid is having 20 carbons, including carboxyl carbon. That statement you have to remember. Fine? Done with this, no? Next. So, first we will see simple lipids. Simple lipids, again there are a few types. First we will learn like neutral or true fats. These are called as what? Neutral or true fats. These are the esters of fatty acids with glycerol. Okay, esters of what? Fatty acids with glycerol. Fatty acids with glycerol. They are also called as what? Glycerides. Based on the number of glycerols, it can be diglycerides or triglycerides. This will be a triglyceride. A glyceride molecule has 3 carbons, each is bearing a hydroxyl group. It has 3 carbons, each is bearing a hydroxyl group. Hence, this is called as trihydroxypropane. Fine. Glycerol, which is trihydroxypropane. This is what? Trihydroxypropane. Fine, right ma? Next. Fatty acids can be of 2 types ma. It can be saturated fatty acids and unsaturated fatty acids. Look into this clearly. Fatty acids could be saturated or unsaturated. Saturated fatty acids, it does not have any double bond in it. Unsaturated fatty acids will have a double bond. Look into this clearly. A fatty acid molecule is an unbranched chain of carbon atoms having carboxyl group attached to the R group. Fine. So, this R group can be again 1 carbon to 19 carbons. What is most important over here is you have to remember the examples. You can remember like saturated fatty acids, so carbons will not have a double bond, unsaturated, so it is having a double bond, this you can remember. But what is most important, you have to remember the examples. For example, saturated fatty acids examples include palmitic acid, which has 16 carbons including carboxyl carbon. Arachidonic acid has 20 carbons. Along with this, you can also take the examples of palmitic acid and stearic acid. Fine. Next, unsaturated fatty acids. Fatty acids with one or more double bonds. What are the best examples for this? Oleic acid, linoleic acid, linolenic acid and arachidonic acid. Many lipids have both. This you consider under saturated only. Yeah. Many lipids have both glycerol and fatty acids. Here the fatty acids are found esterified with the glycerol. Okay. They can be mono, di or triglycerides. For example, oleic acid and linoleic acid. Got the examples of unsaturated and saturated fatty acids, beta? Yeah. Next, many lipids have both glycerol and fatty acids. We have learnt that based on the number of glycerols, we can have diglycerides, triglycerides. So, if one glycerol and one fatty acid, you will call it as monoglyceride. One glycerol and two fatty acids, you will call it as what? Diglycerides. And again here water molecule is released at the time of bond formation. Okay. Next, based on the melting point, triglycerides, can be called as fats or oils. Fats have high melting point, okay, and they remain solids at room temperature. Best example can be your butter and ghee. Whereas oil, their melting point will be low, where they will be liquids at the room temperature. Examples include our ginghly oil and sunflower oil, the oils which we use for making curries and all. Okay. Next, derived lipids. Second type is what? Derived lipids. Derived lipids include steroids. Okay. Steroids do not contain fatty acids, but they include some fat-like properties. That is why they are called as what? Derived lipids. They do not contain fatty acids, but they include some fat-like properties. The most common example of steroids include sterols, that is cholesterol. Common sterol is what? Cholesterol. Cholesterol is most abundant steroid in the animal tissues. Food rich in animal fats contain cholesterol. It is also synthesized in the liver. Fine. Cholesterol is essential component of animal plasma membrane, where 5% of the total composition in animal plasma membrane will be cholesterol. Fine. And then this table is very important, where you have the list of what? Conjugated or compound lipids. Conjugated or compound lipids. Look at it carefully. First is what? Phospholipids. Phospholipids have what components inside it? Usually simple lipids are having what, beta? Fatty acids and glycerol. Okay. But here, including fatty acids and glycerol, any other components are also present. That is why they are called as what? Compound lipids. Here if you see phospholipids, along with glycerol and fatty acids, it also has phosphate group and choline. Okay. Along with glycerol and fatty acid, it also has phosphate group and choline. Best example is our lecithin. Okay. Example for phospholipids is lecithin. Functions include, they form cell membranes, amphipathic molecule. Amphipathic molecule means it has both hydrophilic head and hydrophobic tail, with polar head and non-polar tail. Polar head is what? Hydrophilic polar head. Hydrophilic polar head and hydrophobic non-polar tail. Fine. Next is glycolipids. Glycolipids include fatty acids and alcohol. For example, sphingosine, and it also has sugar molecules, for example, galactose. Okay. Sugar replaces one fatty acid here, and they will form what? Galactose. Fine. They are present in the myelin sheath of the nerve fibers. Okay. of nerve fibers, and chloroplast outer membrane also will have this glycolipids. Glycolipids, the name itself is saying, right, lipid molecules will be having sugar molecules. Phospholipids, the name itself is saying it has a phosphate group. Next is what? Lipids. Okay, here lipids are the main components wherein mainly phospholipid is present along with proteins. This is what? Lipoprotein, sorry. Okay, lipoprotein, lipids and protein. This forms the structural component of membranes, transport lipids to the blood. It will transport lipids in the blood. Last is chromolipids. Chromo means what, ma? Color. Okay. These are colored pigments. For example, carotenoids. These are fat soluble pigments which you see in the plants. Okay. Pigmented lipids involved in coloration and photosynthetic process. For example, keratin and vitamin A. Okay. Take down quickly, ma. Yes. Done? Now. So, these are the images from your NCERT where you have to remember palmitic acid is having how many carbons? 19 carbons, 16 carbons, sorry, including carboxyl carbon. So, here can you see 14 carbons plus 1 15, and including carboxyl carbon it is 16 carbons in total. Here it is glycerol, that is trihydroxypropane. To 3 carbons, 3 OH groups are attached, right? And then triglyceride, 3 R groups are present. See, to carboxyl group, R group is present, forms a glyceride. As such, you can see 3 glycerides here. Fine, this is a triglyceride. And here you can see cholesterol. Okay, this comes under steroids, wherein it does not have a fatty acid and glycerol component, but then it shows some fat-like properties. These come under what? Derived lipids. Okay, remembered. Next is phospholipid, that is lecithin. This comes under what, Nana? Conjugated lipid. If you have NCERT, you take down here only. Cholesterol comes under derived lipids, and this lecithin phospholipids comes under what? Conjugated lipids. Fine. Can you see here? R group is present, glycerol group is present, phosphate group is present, nitrogen group is also present. Fine. Next. Choline is this only. This complete composition is called as choline. Fine. Which has amine group. Nitrogen group is present. This is choline, phosphate group, and glycerides. Fine. Next. So here this chart will give you a gist of complete biomacromolecules we have exercised. First is carbohydrate, which are what are the monomer subunits of this polymers? Monosaccharides in carbohydrates, right? Polymer is, for example, starch. Okay, starch is the storing food material in plants. Next is DNA double helix. Nucleic acid is the polymer of which the monomer is nucleotide. Nucleotide which has sugar, nitrogen base, and phosphate group found in chromosomes or DNA. Next is polypeptide. Polypeptide chain is a heteropolymer of what? Amino acids. Okay, and these are found in intermediary filaments. Next, proteins are found everywhere. We have learnt, like hormones, receptors, channel proteins, everywhere proteins are present, right? In that table we have learnt. Next is lipids. It has fatty acids and glycerol. Triglycerides is the example. Example, adipose cells which has lipids. Fine, right? Next, metabolism. Before going into the enzymatic activity, you have to know on few terms which are given in our NCERT. First is turnover. What is turnover? In a single cell, what all are present? Let us assume a eukaryotic cell. In a eukaryotic cell, what all are present, ma? This, yeah, eukaryotic cell. We have non-membrane bound, single membrane bound, double membrane bound cell organelles. Nucleus is also present, okay, and other cellular components are also present in a cell, okay. always a cell is in a living state means all the components are metabolically diverse. Like in a cell, some things will always be forming, some things will always be destroyed. Okay, that movement will completely be there. When a cell attains equilibrium, it is a dead cell. Yes or no? So that turnover is nothing but all the constituents present inside a cell is dynamic in nature, that is turnover. Okay, wherein one molecule will be forming and the other molecule will be destroyed. So in simple terms to explain, all the chemical reactions that are happening inside the cell, which includes both anabolism and catabolism. Yes. Anabolism includes synthesis of macromolecules. Catabolism includes destruction of macromolecules into simpler molecules, breaking of macromolecules into simpler molecules, right? So this will be what? Catalyzed reactions. All these anabolic and catabolic reactions are enzyme mediated inside the cell. Okay, these are enzyme mediated. Enzyme mediated, which means these are catalyzed reactions. Fine. Proteins with catalytic power are called as what? Enzymes. Means those which speeds up the reactions. Both anabolic and catabolic reactions, those proteins which speed up those reactions are called as what? Enzymes. All the enzymes are proteins, but not all proteins are enzymes. Important statement. Okay. All enzymes are proteins, but not all proteins are enzymes. Next is living state. Just now I told you. How can you assume a cell is in living state? When it is dynamic, all the reactions are happening all the time. If a cell attains the equilibrium, yes, good. The living state, here these are the direct lines from your NCERT, important, look at it. So the blood concentration of glucose in a normal healthy individual will be 4.2 millimolar per liter to 6.1 millimolar per liter, 4.2 to 6.1, fine. While that of hormones would be nanograms per milliliter. See the concentration. Hormonal concentration will be very less when compared to the glucose. The biomolecules are in a metabolic flux. Fine. Just now we have discussed what is metabolic flux. Continuous anabolism and catabolism taking place in the living cell, which we will call it as turnover or the metabolic flux. And then the living state is a non-equilibrium steady state to be able to perform work. Fine. So here you can see, right, all these are biomacromolecules. Okay. After catabolism, they will be releasing the energy, that is ATP, which is the cellular energy, and some chemical wastes are also released like carbon dioxide and water. This is what? Metabolism, and some energy is also released as heat. Fine. Now we will look into the enzymes. Fine. Enzymes, most of the enzymes are proteins, but some of the nucleic acids can also be enzymatic. For example, ribozyme. Some nucleic acid behave like enzymes, for example, ribozyme. Okay. Enzymes are organic catalysts. There are some inorganic catalysts as well, which do not occur in the living cells. Fine. Enzymes are the organic catalysts which occur in the living cells, but those enzymes which does not occur in the living cells, those are what? Inorganic catalysts. Basic introduction of enzymes, clear, right? Enzymes are proteinaceous in nature, quite a few exceptions are there. Nucleic acid can also be the enzyme, example is ribozyme. Ribozyme in mol bio, when we are discussing, we will understand the ribozyme will synthesize peptidyl transferase in formation of a peptide bond between two amino acids. And enzymes are organic catalysts. There are inorganic also, which do not occur in the living cells. Clear? Both are present. Organic catalysts are what? Which are occurring in the living cells. Temperature should be optimum for that. Okay. Whereas inorganic catalysts can occur in outside the living cell. those which catalyze the reactions outside the cell. They can occur at high temperatures as well.
Can enzymes be inorganic?
No. Here you can see the clear differences. Okay, enzymes means organic catalyst only, beta. Fine. Enzymes are complex biological macromolecules made up of what? Proteins. Exception with nucleic acid example. What is that example? Ribozyme. Inorganic catalysts are usually simple, small sized molecules or elements. Okay? These catalysts operate only within the living organisms or biological systems. These are found and function in non-living systems, that is outside the cell. Each enzyme is highly specific to a single biochemical reaction in living, but a single inorganic catalyst can act on multiple different chemical reactions. Here, enzymes lose activity at a temperature above 40 degrees Celsius due to denaturation, but here they remain stable and work efficiently even at high temperatures and pressure. Even at high temperature and pressure they will be stable. Fine, right? Any doubt anywhere, ma? No. Good. Now we will look into the mechanism of enzyme action. What is the enzyme action actually inside a cell? Before that you look into it. As just now we have learned the differences between organic catalyst and inorganic catalyst. The major difference stands with temperature optimum. Yes or no? Here you see enzymes from thermophilic organisms are thermally stable and retain their catalytic activity even at high temperatures, that is up to 80 to 90 degrees Celsius, making thermal stability a key feature of these enzymes. Okay. So, enzymes act as higher temperature inside which organisms? Thermophilic organisms. What will be their temperature optimum? It will be from 80 degrees Celsius to 90 degrees Celsius. Till here it is clear, right, ma? Now we will see two modes of mechanism action. One is lock and key hypothesis, second is induced fit model. Okay. First you see lock and key hypothesis, before that you see this. Yeah. What happens is, first an enzyme, let us assume the example of sucrose. Okay. So, let us assume like inside a chloroplast. Starch is stored as food material, yes or no? After glucose formation, glucose, if it has to be stored, it will be stored in the form of starch. If it has to be transported, it will be transported in the form of sucrose. Remember at least now, glucose after formation, if it has to be stored, it will be stored in the form of starch. If it has to be transported, it will be transported in the form of sucrose. This sucrose, after reaching out any cell, okay, first it will be breaked to what? Glucose and fructose. This will start the process of glycolysis. This is the known story only. This is the known story for us. Sucrose is break to glucose and fructose by an enzymatic activity, yes or no? What is that enzyme, Raghav? Invertase. Okay. Sucrose will be break to glucose and fructose by the enzyme invertase. Fine. Now here you see, what is the substrate over here, beta? What is the substrate over here? Sucrose, right? What is the enzyme? Invertase. So here you see, enzyme will be having the active sites. This is the active site. Can you see here clearly? Enzyme having active site. To this active site, substrate will come and bind. This initially forms out what? Enzyme substrate complex. Okay. This enzyme substrate complex, hydrolysis will happen where water molecule will be released, and this will be formed out what? Enzyme product complex. This forms enzyme product complex, and then products will be released. What are the products over here? Glucose and fructose. This basic model you have understood, no? Now, how the enzyme and substrate are associated with each other, to explain that two major models we need to learn. Okay. One is lock and key hypothesis. Lock and key hypothesis is proposed by Emil Fischer. Emil Fischer, wherein we know how a lock will be and how a key is fit to that. Here the active site of the enzyme is rigid, it is not flexible. According to his explanation, the active site of the enzyme is rigid. To this active site the substrate molecule is fit. Fine. So here if you see, substrate enters the active site of the enzyme substrate complex is formed where the substrate is fit to the active site. There is no flexibility in the active site. Now the substrate is converted to product and product molecules will be released. Okay. Just you have to remember 2 points here. Enzyme is like a lock and key is what? A substrate.
And active site is also rigid.
Yes, it is rigid. It is not flexible. For example, it is of this shape. Okay. Substrate should be at this shape only. This active site is not modulating according to the shape of the substrate.
Yes, but how it will go in with that knowledge?
His model of explanation is that beta.
Yes, but
In reality what you see that he did not explain. He explained one model. Okay. One model of explanation is lock and key hypothesis. According to his model, what is his proposition? That active site of the enzyme is rigid. To that how a key is fit into the enzyme, like that enzyme substrate complex is formed is his explanation.
yes.
Second model is, just a second, okay, no issues. Second model will be your induced key model, wherein enzyme to the enzyme, okay, to the enzyme, substrate will come and bind, yes or no? So here according to induced fit model, which is given by Koshland, which is the most realistic model of enzyme substrate complex formation or enzyme mechanism of enzyme action. This is the most realistic model where he says that the active site of the enzyme is flexible, where it can modulate according to the substrate size. As such, substrate can fit into the active site of the enzyme. Okay. As such, what are formed first? Enzyme substrate complex will be formed, then enzyme product complex will be formed, fine, and then products will be released. Fine. Look at the clear differentiation between lock and key hypothesis and induced fit model. First, this is proposed by whom? Emil Fischer. Sorry, this is proposed by Emil Fischer. Next, this is proposed by Daniel Koshland. What is the actual concept over here? Enzyme's active site is exactly complementary to the substrate shape. But here active site changes its shape to fit the substrate, like how a glove is fit into the hand. Okay. Here key is the substrate which fits into the lock of the enzyme in lock and key hypothesis. Here hand is the substrate which is fitting into the glove. And then flexibility of the enzyme. Enzyme structure is rigid and does not change. Here enzyme structure is flexible and this adjusts to the substrate. I am speaking about the active site of the enzyme. Substrate will adjust into that. See, the place is important where the substrate is coming and fixing. Here in the lock and key hypothesis, according to his explanation, the active site is rigid to which the substrate will come and bind. But here the active site is flexible. For example, this is the shape of the active site according to induced fit model, where the substrate is like this. This can turn to this shape. That is the thing. Okay. Next, specificity. This is highly specific due to perfect fit. This is moderate to high specificity. Fit is optimized after interaction. Okay. And adaptability to the substrate. This cannot explain the enzyme binding to similar shaped substrates, but here this can explain the binding to range of similar substrates. These are the clear differences. and then we have to learn about the transition energy, activation energy, nomenclature of the enzymes, okay, and few set of examples that will be dealt in the part 2, which that is tomorrow, in tomorrow's session. With that, biomolecules will be done. So far from NCERT 11, 2 major chapters will be done by tomorrow, that is 2 major units, that is plant physiology and biomolecules and cell biology, okay. By tomorrow cell biology also will be done. So, that is all for today's class, ma. Thank you.